Mendel's work on inheritance of characters remained unrecognized until the 1900s. There were reasons for the discoveries being undercover for so long. First, his work was not widely publicized because of the lack of communication tools in those days. Second, according to his contemporaries, Mendel's concept of factors controlling heredity could not explain the continuous variation seen in nature. And third, Mendel used mathematics to explain the various permutations and combinations in his experiments. This mathematical approach was not acceptable to his contemporaries. However, Mendel's work was rediscovered in the 1900s. By 1902, chromosome behavior during meiosis was unveiled. Walter Sutton and Theodore Boveri independently found a parallelism between the behavior of chromosomes during meiosis and the transmission of Mendelian factors. They explained this by the chromosomal theory of inheritance. According to chromosomal theory of inheritance, the units of heredity, that is, the genes, are located on chromosomes. This theory was developed as a result of cytologists examining the behavior of chromosomes under microscope and geneticists studying the behavior of genes. Following are the salient features of the chromosomal theory of inheritance. The somatic cells of a diploid organism possess two sets of chromosomes. One set is received from the father and the other set from the mother. At the time of meiosis, during gamete formation, chromosomes of a homologous pair separate. Chromosome behavior during meiosis indicates that genes are located linearly on chromosomes. Sutton and Boveri found out the following parallels between behavior of chromosomes during meiosis and behavior of Mendel's hereditary factors. The somatic cells of an organism have two identical sets of chromosomes. This means that the chromosomes occur in homologous pairs in diploid cells. Mendel also proposed that the hereditary factors exist in pairs in organisms. During gamete formation, the chromosomes of each homologous pair segregate in meiosis 1. So each gamete receives one chromosome from each homologous pair. Mendel's law of segregation also proposed the same thing, that the paired factors segregate during gamete formation. During meiosis, the chromosome pairs segregate independently of every other pair. According to Mendel's law of independent assortment, the factors in each pair are distributed into the gametes independent of the other pairs. When the male and female gametes fuse to form the zygote, the diploid number is restored because the homologous chromosomes come from both parents, one set from each of them. Mendel had also proposed that the paternal and maternal characteristics mix in the progeny. Whether the characteristics they carry are expressed in the individual or not, the chromosomes retain their individuality throughout the life of an individual. According to Mendel's observations, the factors are not lost even if they are not expressed in the progeny. Based on all these observations, it was concluded that the behavior of chromosomes was similar to what Mendel had described for the behavior of factors. This led to the formulation of the chromosome theory of inheritance. The first evidence of the chromosomal theory of inheritance came from the experiments of Thomas Hunt Morgan on Drosophila melanogaster, or the common fruit fly. Drosophila is easy to grow. They have a very short life cycle of about 2 weeks. They show a clear differentiation of sexes as male and female, and show a large number of hereditary variations that can be easily observed under low-power microscopes. Morgan's experiments on Drosophila and his experimental verification of the chromosomal theory of inheritance helped explain the variations produced in sexual reproduction. It is now proved that genes are located linearly on the chromosomes. With progress in molecular biology, we now know that a chromosome is made of a molecule of DNA, and segments of DNA are the genes.